So today, it's a great pleasure to introduce Dr. Emma Thomson. Emma is an MD, PhD. She has been trained in the UK, in London, both for the clinical activities and for the scientific activities. She got her PhD at Imperial College, then a postdoc at Oxford. She is now a clinical professor of infectious disease in the London School of Hygiene and Tropical Medicine, but also the head of an MRC Research Center for Virus Research. She has also been very much connected to public health, being a consultant to WHO. She's a member of a number of very interesting groups organized in the UK to share the results regarding variants, such as the UK Variant Technical Group, COG-UK consortium, and others. And so she's very much involved in networking and cooperation. And last but not the least, she's a GVN member. And so we really welcome her lecture today, which will be entitled Characterizing the Omicron variant of SARS-CoV-2. Emma, thank you again for giving this lecture.
Thank you very much for the introduction and the invitation to speak today. It's a great pleasure. I'm just going to share my slides. Okay. Can you, just to double check that you can see these and also you can hear me clearly?
Absolutely.
Okay, thank you very much. Yes, so I'm going to talk today about some of the work that we've been doing in the UK to characterize the Omicron variant of SARS-CoV-2, and also I'll give a bit of background in the work leading up to this as well, which has largely been as part of the COG-UK consortium, which was led and has been led by Sharon Peacock in the University of Cambridge and the UK Health Security Agency. I thought, first of all, since this is a global network meeting, that I would introduce our centre. This is the MRC Centre for Virus Research in Glasgow, and we have these buildings here and here, and quite a reasonably large centre now with 21 laboratories working on all sorts of viruses. And back in March 2020, we decided that given the impending pandemic, we would really switch off most of our other activity and concentrate on SARS-CoV-2 as it emerged across the world. And we've now gone back to probably working around 50-50 or back on other viruses as well as on SARS-CoV-2. A lot of the work that I'm going to present is as a result of a large sort of cooperation between multiple laboratories, 16 main centres across the UK, of which we are one of the most northerly here. And we've sequenced well over 2 million SARS-CoV-2 genomes as it emerged in the UK, and this was because there was an investment put into sequencing quite early on in the outbreak, and we then used that to catalyse some further research investigating the phenotype and the clinical presentation of SARS-CoV-2, and we got increasingly involved with data linkage. And I think the power of genomics is really much increased by being able to associate critical clinical data and epidemiological data, as I'll go on to sort of illustrate, with the sequence data. And it has actually also been implemented within our public health agency systems, which I think is has been a significant advance for us. So as I mentioned, way back in March 2020, COG-UK was formed, and we started to use sequencing as an early warning system in the UK, using a local approach to sequencing. So multiple laboratories were involved. In Scotland, which is where my responsibility has been, we sequenced between two laboratories, the 5 million population of Scotland, and also have been involved subsequently with linking all of that information with the health records from people from across the country. And I thought I might just, I guess, since it's been 2 years now, illustrate some of the first sort of useful findings that I think we had from the sequence data. There was at the very beginning some doubt about how useful sequencing would be, what the utility would be, and one of the first papers that we were involved in was the second one listed here, and then my colleagues from Oxford and Edinburgh and elsewhere were involved at a UK level at estimating how many times the SARS-CoV-2 virus entered the UK in the first month of the outbreak. And in Scotland, this happened around 300 times, and in the UK as a whole, more than 1,000 times. And this just, I think, illustrates how quickly the virus emerged, and the likelihood of another respiratory pathogen coming into the country would be very likely to emerge at the same levels of high frequency if present in countries in which we have very strong links. And we worked on characterising in Scotland where the introductions came from, and unfortunately at the time, and I was working on the wards at this time in the first wave, we were really only testing people initially from China and subsequently from Italy, but all of our importations in fact were from Central Europe, where we have most linkage and with international travel, and this was very closely related to the sort of flight paths that are present between the UK and other countries. And so this just simply illustrates here the first 4 weeks of the outbreak as it emerged in Scotland, and in different colours and across the x-axis here you have the different days of the week. And initially we were importing SARS-CoV-2, probably mostly from Italy and subsequently Spain. We could tell that from the genomic data, but we had very rapid community transmission, as shown in red in this diagram, and much more rapid than we had anticipated would happen. We have subsequently seen exactly the same phenomenon again and again, and so we actually looked at the second wave, and this followed quite a strict lockdown in the UK, so much so that the virus almost was extinguished. And if you look at the lower panel here, you can see these are the different lineages that we had in Scotland at the time, and these vary in colour. The deeper the colour here, for example, these are the more dominant lineages. And you can see that these largely became extinct over time and were replaced after the first lockdown with a whole batch of new lineages of infection. And I think this told us something about the risk of high frequency importation and our interconnectedness with the rest of the world. And of course, that has happened with every lineage since, other than Alpha, which appeared to originate in the southeast of England in Kent. And I'll come on and talk a little bit about that before moving on to the sort of ultimate virus that we've, the ultimate variant, if you like, in terms of immune evasion that we've reached now, which is Omicron. We also used genetic data to track infections as they occurred in hospital environments, and one of those hospital environments where we had real problems in the UK was in renal dialysis units. And what we were able to do with this study, and I think this is important in terms of thinking about setting up such analyses as routine public health investigations, was to look at the interconnectedness of sequences from people who became infected who were attending renal dialysis units. Renal dialysis units, of course, could be considered to be a conduit between the community and the hospital, because people spend 3 days a week, 3 half days a week in the hospital environment, and then on hospital transport in the community. And what we did here was, I think, something that we certainly had never done before, which was to link the genomic sequence data, which gave us a certain amount of signal, but the signal at this stage was not perfect. And as many people on the call will know, there really wasn't that much variation in the genome in the first few months of the outbreak. And so we actually linked the genomic data with epidemiological data. For example, what days people dialyzed on Mondays, Wednesdays, and Fridays. And we linked it also with their home location, their zip code or postcode. And we found that we made a tool which would help us to predict how likely it was that somebody was infected in the renal dialysis unit, in the hospital, or in the community. And if you look in this middle section here, UK 429, you can see that, there were some very closely related sequences here, and these actually were quite unique to the hospital environment and did not reflect the viruses that were circulating in people's home residence. And so this score, first score here, was a score assigned with likelihood of being infected within the renal dialysis unit itself. However, other closely related sort of sequences were much harder to assign to being a hospital transmission, because actually, for example, this lineage here, there were 63 other very similar lineages, many of which actually occurred in the home locations of those people who'd become infected. So it was a mixed picture of people being infected, both in the hospital and in the community. But combining the sort of epidemiological data along with the genomic data helped us to figure out what was going on. And then this early warning system that we have in the UK has now really been pivoted almost entirely to immune evasion. We can also estimate things like growth rates of different lineages, but the immune evasion is really key, and we need to understand whether or not our vaccines are likely to hold up, and we can predict that to a certain extent from the genomic data. The other thing that I think that we were not able to tell from the genomic sequencing, and I'm going to come to that in a minute, in particular about the Omicron variant, was that there was actually a fundamental shift in the biology of Omicron, which really wasn't something we could have predicted from looking at the sequence data, although going forwards, we may be able to do that because we've started to be able to map the determinants of cell entry onto the S2 segment of the spike protein, and that probably determines whether or not a virus will enter by endocytosis, as I'm going to come on to describe shortly, or through the direct route via TMPRSS2. But just going back historically to what we first found in the sequencing data, one of the first mutations that was described was D614G, and of course this became global, and Eric Volz at Imperial College did some lovely work to show that this was associated with an increased growth rate and increased transmission. However, this was not associated with an increase in immune evasion, and I guess at that time the pressure on the virus was not as high to evade immunity, because most of us were susceptible at this point. But it was clearly adapting to become used to life in human cells. Then again, early in the pandemic, we noted a single polymorphism, and I thought I might just describe this just to show how we kind of started to understand through the sequencing data that certain changes in the genome might be associated with immune evasion. And the first one that we spotted, and I think this was one of the very first immune evasion single polymorphisms that was described, and in fact it's not anything like as important as some of the others that we now see, but this N439K variant came up, and it was noted to be in the receptor binding motif of the receptor binding domain, and so it interacted directly with the ACE2 receptor. And this change, we had some help from collaborators at Beer and the University of Washington, and we were able to show that this change, N439K, was associated with the formation of a new salt bridge, which increased the binding affinity of the receptor binding domain to the ACE2 receptor. And this is often a feature of mutations that we see now in the new variants, which I'm about to come on to describe. This very first mutation that we saw originated in Scotland. We spotted it in NHS Greater Glasgow and Clyde, where I'm based. And actually, after that first lockdown that I mentioned, it became extinct. And so if you remember those lineages, this one disappeared. But and so we kind of became less worried about it. But then we noticed it popping up in Central Europe, and some countries in Europe had quite high prevalence for a while of this variant. So Ireland, in particular, the Republic of Ireland, and also other countries, including Denmark and Romania, we kind of spotted this variant popping up in. And it was re-imported back into the UK as well. And so we had it around for a while, and it clearly was giving a bit of advantage to the virus. And we did some workup of this, and we've kind of used some of these methods with the new variants to monitor and track how they are behaving as well. So we were able to show that the D614G, in fact, N439K always occurred with D614G, was associated with a lower CT value, and we were careful to make sure that we looked at actual viral load rather than just CT, and it was associated with high viral loads or low CT values. And actually in culture as well, it appeared to have a kind of higher takeoff in cells co-expressing TMPRSS2, and I will talk about TMPRSS2 again shortly. And we used cross-competition of N439K and 439N variants to see if they could outcompete each other, and they were at least as fit. So N439K was at least as fit as 439N, if not a little bit more in TMPRSS2 expressing cells. I don't think we appreciated fully what TMPRSS2 was doing at that time, actually. And then we set up a system for monitoring whether variants were associated with severe illness. And I think this has been quite important. And I think this linkage of data is critical, and systems for data linkage, ideally on an international scale, would really help us understand more quickly how dangerous a variant is going to be and how we will need to react quickly to it. So we created a scale from 1 to 4 with no support required, oxygen in hospital, either invasive or non-invasive ventilation, and death. And of course, we saw this pattern with very high mortality and hospitalization, people who are older, but we didn't see any difference between those people who had an N or a 439N or a 439K. We did, however, spot some preliminary evidence of immune escape. So IgG binding to receptor binding domain protein, which had K, was reduced in quite a lot of people who had their serum screened who'd been infected. If people had been infected with a 439K variant, then the neutralization and binding, both binding and neutralization, was pretty steady, indicating that what you're exposed to really matters in terms of your immune response. And we also showed that there was actually a decrease in binding to the Regeneron, one of the Regeneron antibodies, 10987. So this really heralded, and these were the first things that we were observing, these polymorphisms, but this obviously heralded this very rapid evolution of the virus that occurred in variants. And as you know, the new variants have had stepwise evolution with multiple mutations arising all at once. And the first of these was Alpha, spotted in the southeast of England and Kent, and then followed rapidly in the UK anyway by Delta, and subsequently Omicron. And with each one of these sort of emergences of these very successful variants, we've seen a gradual accumulation of changes in the spike protein, such that Omicron, and this is the BA1 variant of Omicron, has an accumulation of a very vast number of changes within the spike. And around half of these were polymorphisms that we recognized through our early warning system as being associated with immune evasion. However, some of them we didn't really know what the function would be, and I'll go on to talk about that a little bit later. So However, the concern in terms of such obvious variation in antigen, sort of the antigenicity of Omicron, really alarmed us, and we were quite concerned about this, the emergence of this variant, and so quite rapidly used sera from the deployed recipients of deployed vaccination, which was rolled out relatively quickly in the UK. And we looked at people who'd had 2 versus 3 doses of vaccines. And you could see overall, firstly, and this work was done by Brian Willett, who has been running the serology response at the MRC Centre for Virus Research. And what you can see here is that the 3rd dose sera resulted in quite a significant drop in neutralization in all of the Omicron variants that we tested, BA1, BA1.1 and BA2. There is a suggestion perhaps that BA2 may be a little bit even lower than BA1, but it's very subtle and certainly doesn't reach statistical significance. However, it may be more significant at the kind of monoclonal level, and we have tested also monoclonal antibodies, and I'll just show that in a second. Some of the participants in the DOVE study, our deployed vaccine study, had matched samples following dose 2 and dose 3, and I should say that these participants were balanced in age and had a median age of around 50, and so these are the sort of plots of neutralization in people who'd had 2 or 3 doses of vaccine, and you can see that with dose 3, perhaps in this figure panel here, C, there was almost always a sort of nice increase in neutralization of Wuhan. The responses to the Omicron variants were lower to start with, and did have a bit of a boost, but these results were quite heterogeneous, and these lines just illustrate that heterogeneity in responses. And when you look at monoclonal antibodies, this is Ronapreve here, which of course we've mostly stopped using these days because Omicron is not sensitive to it at all. So tropomab is what many of us have been using in clinical practice, and it's a sort of harder one to look at with the pseudovirus system, but we did see a significant drop in neutralization of BA2 versus BA1, and so it probably is less susceptible to sotrovimab than BA1. We next looked at T-cell responses in Omicron, and these were done using peptide sets which come from the PITCH consortium led by Susie Dunachie and Paul Kleinerman at the University of Oxford, who kindly gave us the peptides and advised us on the protocols. And really, I think this is to be expected, that there wasn't really a huge sort of difference between Omicron and Wuhan peptides in terms of response. One thing that I think is noticeable is that the Moderna recipients, so we looked at the different sort of vaccines that were used in the UK, either Pfizer, the Oxford AstraZeneca ChAdOx1 vaccine or Moderna vaccine, and there did seem to be a slightly higher sort of response in those recipients of the Moderna vaccine, but there was no significant difference between those who the peptide response to peptides from Wuhan or Omicron, and that is probably reflected in the fact that there is some loss of, there's some change within known T-cell epitopes, but only about 28% or so are affected. So to pair this up with actual vaccine effectiveness, so we looked at neutralization that I've just showed you just now, but obviously that doesn't always exactly replicate what we see in real life. We have a system set up and a study called Evade, which is funded by HDR-UK. And this HDR-UK award was really set up to allow us to be able to link better clinical data within a safe environment. So we have this sort of trusted research environments that allow us to do this sort of work. And this shows the rollout of vaccination in the study area in Scotland. And these are the sort of different doses of vaccine at any time point. And then this second panel shows the variants that we saw over time. And so initially this was largely B1.177 and other B lineages, and then taken over by Alpha in green, Delta in purple, and Omicron in pink. And we looked at test positive results, positive and negative results, to be able to estimate vaccine effectiveness, which is shown in this slide. So if you haven't been previously infected or documented to be infected with SARS-CoV-2, and you've had 3 doses of vaccines, then there is a significant drop in vaccine effectiveness against Omicron versus Delta. But what's useful to know is that that 3rd dose makes a huge difference. So if you look at people who've had 2 doses, there's a huge lift in the vaccine effectiveness after the 3rd dose. And I think importantly, we separated out those people who had actually been infected. And I think this part of the second panel here is quite important, that people who have had an infection and 3 doses of a vaccine present really quite a high barrier to reinfection, and vaccine effectiveness is really quite high even against Omicron. And with the same population, we looked at clinical severity of disease, and I described this before from when we just giving you the background, when we looked at single polymorphisms and tried to link up severity and different variants. This time we looked at we've looked at the transitions of all of the variants over time, including Delta to Omicron. And importantly, these come from windows where both variants are at least at 5% of the mix. And so factors like treatment and so on shouldn't be hugely influencing outcomes. And what we found was that the transition from B1.177, which was one of the B lineages dominant in the UK before Alpha, was associated with less severe illness than Alpha itself. And you can see this across the different age groups. And again, illness was much more severe in older people. And in the first wave, we had very high mortality rates. We didn't have such good treatments and so on. And you can see that in every one of these groups, the severity, people who died or people who were hospitalized is higher in Alpha versus the preceding variant. The same thing happened again with the transition from Alpha to Delta, and so the virus actually appeared to be more virulent in people who were infected with Delta versus Alpha. There was no difference between Delta versus AY4.2, which was a sort of Delta plus dominant variant that we had in the UK. And then the very opposite thing happened when Omicron arrived, and we started to see a decrease in clinical severity associated with Omicron infection. And this really is where we got lucky, I think, with this virus, because it's very immune evading, as I've just shown. But thankfully, the clinical severity, while not insignificant, and there have been deaths, particularly in older people, and hospitalizations, the clinical severity of disease is much reduced in Omicron versus Delta. And then as we were doing this work, and I'm just going to show a few slides, and a lot of this work comes from the laboratory of Joe Grove at the MRC Centre for Virus Research. We kind of all joined together on this paper. We noticed that the virus was behaving differently in culture. And so what you can see here is in panel A is syncytia formation. And we used a split GFP system, which involves the expression of different parts of the GFP protein in different cells. And when these adhere together, as happens with when you get syncytia formation, you see this sort of green fluorescence. And so you can see here with Delta, a lot of syncytia formation and fluorescence, a little bit less with Wuhan, and then it almost completely disappears with Omicron, indicating that the cell-to-cell adherence, which is a feature associated with pathogenesis, was much reduced. And you can see in the right-hand panel here, the fluorescence sort of mapped. Now, we went on then to see what happened in, for example, different cell types. And you can see here an infection of Calu-3 cells that in panel D, that Omicron grows less well. And we wondered if that was because of a difference, that alongside the syncytia formation data, we wondered if that indicated a change in the entry route of Omicron. And indeed, that's what we found. And so there are two potential routes for coronaviruses to get into cells, and most of them get in through endocytosis, what we've referred to here as route 2. But SARS-CoV-2, all the previous variants, Wuhan variant, Alpha and Delta, they get in through this interaction with TMPRSS2, which allows breakdown of the spike protein into the different spike components, allowing and promoting cell surface fusion. This can be inhibited with an inhibitor called camostat. And the alternative pathway, which occurs with other coronaviruses, and including the pangolin coronavirus, is to enter cells through endocytosis, and that can be inhibited by E64D. And what we found was there's a differential between Delta and other variants and Omicron in getting into different cell types. So Calu3 is a lung cell line, and Delta gets in really well, but Omicron less well, and pangolin, which relies on endosomal entry, even less well. And Calu3 cells express a lot of TMPRSS2. We then looked at HEK cells, which promote the endosomal route of entry, and you can see here that Omicron has a significant advantage, as does pangolin CoV. The A549s that a lot of us use with ACE2, co-expressing ACE2 and TMPRSS2, which allow both routes, you don't see such a difference between any of these different variants of the entry. So we then went on and carried out inhibition experiments, and we were able to show that Omicron can be, the entry of Delta can be inhibited by camostat, and the entry of Omicron can be inhibited by E64D, demonstrating basically that that entry is mediated through largely or preferentially through endocytosis. And you can see that there's a dose-dependent relationship here with these inhibitors. And on Western blotting, you can see that there are more that you can get downstream products with Delta and TMPRSS2, but not the other way around with Omicron. And so basically, this indicates that there has been a very fundamental change in the entry biology of Omicron versus Delta and the other variants. Actually, the cell surface fusion can be overcome by adding trypsin, so that allows to protease, which will allow will take over from the absence of TMPRSS2. So then we went on to, or Joe and colleagues went on to look at the different domains of the Omicron spike protein that might be defining this entry process, and swapped around using the pseudovirus system, so not using live virus, but pseudovirus, swapped around the NTD receptor binding domain and the S2 domain of Omicron into the Wuhan background. And what you really see here is that Wuhan with the Omicron domains get in through, that really increases the endosomal entry in HEK cells. Whereas if you have Omicron with Wuhan domains, it is inhibited. And so S2 appears to be a key determinant of this entry process. And similar experiments with the inhibitors that I discussed before shows that these can inhibit this entry process as expected. And I think, and really just finishing now, but I think that these messages and these sort of with these chimeric viruses is something that we need to think about quite a bit, because of the emergence recently of many recombinants. Perhaps the most concerning of these are the recombinants which are made of Delta versus BA1, but there are many also BA1 and BA2 recombinant viruses. Now, XD, which was found in France, and there's some nice data emerging on this, has the spike of Omicron and the background of Delta. I think I would be really a lot more concerned about a virus which had the backbone of Omicron and the spike of Delta, given the changes in entry and how that may define clinical severity of disease. But we will need to, I think we can't stop learning about this virus, and we will need to carefully monitor as these recombinants emerge, whether or not they are associated with severe disease and immune escape as well. So I think that's the end of my presentation. I just in summary would like to summarize to say that I think genomic sequencing has been very valuable as an early warning system to estimate things like transmission patterns and growth rates. And I think its role is very clear in terms of monitoring for antigenic evolution. And I hope that we will be able to continue to do that, although I think in the UK this will be at slightly lower scale given the lower pathogenicity of Omicron, but we don't know for sure, also given the heterogeneity in clinical severity of disease, that the next variant may not be more severe. And I think one of the ways in which we might monitor for that is to look at the way that future viruses enter cells, and also whether or not they are involved, they are associated with syncytia formation. And a change in cell entry of the virus was actually something that was not initially predicted by genomic surveillance. And so the fundamental virology is also critical. But we should now be able to know a little bit more about changes in S2, which might determine whether or not virus can have this direct entry into cells, which may be associated with more severe disease. And I'll stop there. Thank you very much. And I should say, we've had a lot of funding from the COG-UK consortium, the Genotype to Phenotype Consortium, HDR UK, the Medical Research Council, and others. And this is really reflecting the work of very many people, and I'm presenting on their behalf. Thank you.
Thank you so much, Emma. This is really, that was a great lecture and a lot of information and such an illustration of what you can get from consortia and networks and sharing data and resources and modern technology. Really great, great lecture. So we already have some questions on the Q&A. Can you see them? Can you read them?
Yes, I can.
Please.
Yes. I might start in reverse order with the BA2 because I know more about it than BA4. So BA2 enters cells in the same way as BA1. So we've been able to show that it also enters through endosomal entry, and it also is associated with very low amounts of syncytia formation. And so it's quite similar in that regard. It does seem to be a bit different, of course, in terms of its immune evasion capacity, but not significantly. It's not a huge jump in terms of neutralization. It also has become dominant from BA1, and so it must have advantages. I am not sure exactly yet what those advantages are, but the immune evasion, there's perhaps a slight advantage. And BA4, we haven't looked at things like neutralization entry with BA4. I think that we would expect that entry would be very similar, and BA4 has now been split into BA4 and BA5 actually, which are very closely related, and we will have to wait and see really what we find in terms of severity and immune evasion with these variants. I do know that it does look like BA4 goes back to November last year in terms of dating, and so it was probably present around about the time of the emergence of the other BAs, and it's just that we detected it fairly recently, so it's quite diverse and indicating that it's been around for a few months.
Thank you. You have another question.
Okay, so this one is anything revealed by ages of persons infected, vaccinated, reinfected or not? So certainly we know that people who are vaccinated have better response. The younger you are, the more durable your immune response is to pretty much any variant of SARS-CoV-2. And so there is an age-related component, and we always include that in our modeling. people who are older, their immunity is likely to wane more quickly. And of course, people who have other types, so people who are immunosuppressed for any reason will also have less powerful neutralization responses, or even absent neutralization responses following vaccination. There's a few more here. Okay. Yeah. So there's a question here about the endocytosis pathway. Does this make the Omicron variant even more resistant to neutralizing antibodies than predicted on RBD changes because of less accessibility to the endosome to antibodies? Yeah, it's a really interesting idea. I guess it depends how long the virus is present. If it is hiding inside the endosome, I guess it is an interesting idea. I don't think it's something that I can answer, but it's I guess that's possible. And then, can you already say anything about the pathogenicity of XE? So I can't say anything based on data, but it does look like, so XE is BA1, BA2 variant, and I don't think I would expect to see a significant difference in the pathogenicity of XE. I think that largely will be, sorry, my blinds are, hopefully you can't hear them, they're blowing around. I'm just going to try and get them in. The pathogenicity of XE will probably be determined largely by spike. So I think that, I'm not expecting to see an increase in severity with XE, but then again, we have been repeatedly surprised by this virus, and I think we do need to look. XD, I might be a little bit more worried about than XE, just given the fact that it's Delta with it's Delta with the Omicron spike, so that is probably more concerning. And just to slightly. So XE has BA1, and then it's the break point is well ahead of spike with BA2, and so I don't think we would expect any major change in pathogenicity with that. But I hesitate, because every time we say something definitive like that, we find something different in the data when we look.
Thank you very much. That's so I believe that we have we had the different question. So that's great, and we have this COVID-19 task force, and we hope that you can join this task force with all the information that you are providing. And so again, we will conclude now. It's time, but congratulations. Thank you for everybody to attend. Thank you, Emma Thomson, for a great presentation. And we wish you, as always, very good day, evening in this GVN network. Bye-bye. Thank you so much.
Thank you very much. Bye-bye.